Genetics. What can it mean? The ability to perfect the physical and mental characteristics of every unborn child. Of genetic engineering.
To choose the genes.
In the sense of society. CRISPR is a new gene editing tool that can be used to change genetic sequences in living organisms, giving it a huge potential in modern-day treatment of disease. However, controversy exists around its use, due to many ethical implications which we will describe in this video. We aim to explain the history and mechanisms behind CRISPR-Cas9 as well as outline the positives and negatives of technology to allow you to come to an informed decision of its use.
We will be using the following key terms. DNA, the molecules carrying genetic information used for protein coding. RNA, a one-stranded messenger molecule used to transfer DNA information for protein production. Enzyme, a protein used to speed up or initiate a chemical reaction.
CRISPR stands for clustered regularly interspaced short palindromic repeats and was originally used by bacteria. While this sounds complicated, all it really is a segment of DNA containing short repetitions of sequences. In this kind of genetic engineering, a CRISPR sequence and its associated protein is used to edit the arrangement of bases in a gene.
Some viruses use bacteria as factories to replicate by inserting their own genetic material inside them. This usually kills the bacteria, but sometimes they manage to survive using their own antivirus system. The surviving bacteria save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. It is stored safely in here until it is needed. When the virus invades again, the bacterium quickly makes an RNA copy from the DNA archived and arms a secret weapon, a protein called Cas9. This scans through the bacteria for any signs of the virus invader by comparing every bit of DNA to the archived piece. When it finds a match, it is activated and cuts out the virus DNA, making it useless. This protects the bacterium against the attack.
An example of what CRISPR can help solve is sickle cell anemia, a condition where the blood does not carry enough oxygen around the body. This is caused by a mutation where a T base is swapped with an A base.
The CRISPR technology can reverse the A to a T using the Cas9 enzyme and a piece of guide RNA. This guide RNA is complementary to the DNA sequence responsible for the sickle cell mutation.
The Cas9 enzyme and guide RNA attach to both strands in the target DNA sequence and cut them, acting like a pair of molecular scissors.
The gap can then repair via natural mechanisms, but the wrong base may be inserted. Scientists can design a small piece of DNA to fill the gap with the correct sequence, improving the likelihood of a successful alteration.
A huge potential positive of CRISPR-Cas9 is in cancer therapeutics, due to the number of ways in which it can be used to combat the disease as a result of its gene editing function. These potential methods include using CRISPR's gene editing function to correct mutations in the cancer cells responsible for rapid cell reproduction, or knockout genes essential for the survival of the tumour. Using CRISPR for viral gene editing, such as disrupting the genes in cancer-causing viruses such as human papillomavirus, a virus responsible for causing cervical cancer. Genetically editing other human genes, such as enhancing tumour suppressor genes or improving the ability of the immune system to recognise and kill tumours. This technology also has the potential to treat genetic diseases, such as cystic fibrosis, by altering genes involved in the diseases during the embryonic development stage, preventing the disease onset in later life. CRISPR-Cas9 is also already being used by drug companies during drug development. One current use of the enzyme is to genetically alter mice so that they can contain human genes, meaning drugs tested on these mice have already been tested on human genes. This process may even remove the need for human drug testing entirely in the future.
CRISPR-Cas9 marked a milestone discovery in the future of genetic engineering and modification. However, the risks of the groundbreaking technology provide a cause for concern. With the potential to wipe out whole DNA sequences and alter the characteristics of an organism, CRISPR can be considered a frightening concept. Because some sections of DNA are very similar, the CRISPR technology may target the wrong section, causing off-target mutations and potentially devastating effects on the genome. Other fears surround the potential of CRISPR technology to be exploited for use as bioweapons. In theory, CRISPR will be able to modify the characteristics of organisms, such as mosquitoes, to produce harmful toxins or other hostile properties. The modification of human sex cells to produce designer babies is also of great concern. Where will the line be drawn when gene editing becomes available to exploit? Though the ability to control genes that can cause genetic diseases would be beneficial to many, the grey area surrounding the ability to control athletic performance, intellectual capacity and aggression, to name just a few, remains an ethical issue.
CRISPR-Cas9 is becoming extremely relevant in modern-day medical science. In this video, we have outlined the potential positives of the technology, as well as current ethical concerns. We hope that by watching this, you can come to your own conclusions whether or not you think CRISPR should be used in modern medicine.